This talk will be a slight whirlwind tour, but thankfully this is recorded so you can go back and pause whenever you'd like on the HBM website. So my name is Jesse Gomez. I'm a PI here at the Princeton Neuroscience Institute, where I run the Brain Development Lab. And part of our work in understanding how the brain develops is to link changes in function with those structural changes occurring at a much smaller scale. So we'll go over 3 vignettes detailing how you can link structure and function in the human brain. And hopefully there's enough code here so you can go forward and do your own discoveries. So this will be a sort of cooking with Martha Stewart style thing. I said, well, of course, we'll be doing neuroscience, and hopefully I'll show you how you can take pre-processed selectivity maps or other cortical maps you might have, and using some data sets or pipelines, be able to link them to anatomy, genes, or proteins across the brain using code to discover your own structure-function relationships. So we'll start with how you can link gene expression across the brain with functional representation. So as many of you likely know at this point, the Allen Institute has produced an atlas of the adult brain detailing the expression of close to about 15,000 unique genes across the brain, and when pooled across their 6 donors, which you can see here, this measurement of gene expression covers most of the brain, including some subcortical structures. And in this data set, they extracted a tissue sample from a given brain region shown in the upper left in the brain there. They process it and perform a microarray analysis to quantify gene expression through RNA content. And from a single sample, they can quantify the presence of many different genes. And each green dot there in that little green matrix is one well within a microarray analysis, and it fluoresces based on how much the RNA content is there for a given protein of interest. So from a single sample, they can measure up to 15,000 different RNA transcripts, and they map these RNA transcripts back into the native brain space of each individual, and each brain is an MNI space. So you can pool these 6 donors in MNI space and relate them to your cortical map of interest, which I can show you how to do here. So we'll detail an analysis pipeline that should pretty easily let you take any brain map you may have and see how regional variation in your map of interest might be related to variation in certain genes expressed within the brain. So this sort of analysis can be rife with pitfalls, and since the release of the Allen Brain Atlas, a gold standard for how to process and analyze expression data has emerged. And thankfully, this toolbox will perform that gold standard analysis for you, in addition to some nice data visualization. So this pipeline is from the paper shown in the upper left there. So if you want more details, of course, you should look that paper up. But the first step shown here in this little cartoon terminal window is assuming you have Conda installed on your computer, which is a Python environment, is to create and activate a space for this toolbox, and we'll call it transcriptomics. This should only take a few seconds. Next, in terminal, you'll need to use git to clone a toolbox called Enigma. For this package to run some of its statistical analysis, you'll need this toolbox. So once you have it cloned, we'll change directories into that folder and run Python or Python 3 to set it up. If it gives you any grief about a toolbox missing, you can use pip to install that toolbox and then try this step again. Again, you're going to use pip. You're going to download another toolbox, which is at that link there. You could simply copy and paste that to install that last toolbox. And now that all this groundwork is laid, the last thing we'll want to do is install the actual imaging transcriptomics toolbox itself. So once all of this is done, we can go ahead and get started. And running this toolbox really is as simple as a single command line, which I'm showing you at the top of the screen there. And each of the different inputs for the command line or the arguments for the command are shown in different colors, and we'll walk through them now. So probably the most difficult one to get is going to be the input in blue here. This is just going to be a contrast map or a selectivity map in an MNI space. So you might originally, from your MRI experiment, have a T1 weighted anatomical image of your subject's brain, and you'll have a contrast map from your experiment aligned to that anatomical image. And for the sake of this cooking show together, let's see if we can find any genes that might be correlated with face selectivity across the brain. So once you have your T1 image and your contrast map in the same space and same resolution, the next step you'll need to do is transform them into MNI space. This can easily be done with the SPM GUI in MATLAB. If you have SPM path in MATLAB, you just type SPM fMRI and it'll open that GUI. And there you can select, normalize, and choose your subject's T1 weighted brain image, and it will use that T1 image to align to MNI space. And the nice thing about SPM is that it lets you apply that transformation to any number of additional images. So you'll make sure you'll want to select your contrast map, in this case, the face selectivity map, so that also ends up an MNI space. Next, it's slightly annoying, but this toolbox uses the old field of view size from MNI, and in order for it to run, you need to run one more line of code to reslice your anatomical and contrast map to have a field of view of 182 by 218 by 182. And the easiest way to do this is to use a reference volume that already has that field of view. Luckily, the paper that I showed you on the previous slide provides a reference volume for you to use. And once you get that reference volume, it's as simple as running that FreeSurfer command down there, MRI convert. You give it the input map, which is just your contrast map, the output, which is just the name of the contrast map once it's been resliced, and then you give it the RL flag, which stands for reslice like, and you feed it the reference file, which has the dimensions shown there. So now that your contrast map is in MNI space with the appropriate field of view, you now have your input file represented in that blue text above. So you just make sure you feed it the full path to this new contrast map in MNI space with the new field of view, and then we can move on. The output is very easy. You just give it a full path to the folder where you want all the output stuff to be saved. The next argument is the regions argument. This is very easy. You can tell it all or cortical or subcortical. In this case, we're just going to use all of them, but all means that it includes all of the cortical and subcortical labels from the Desikan-Kiliani-Tourville Atlas, or DKT Atlas. This atlas, like any atlas, is specific. It may not be perfect for what you want to do, but I'll walk through what the code does with an atlas at the highest level so you can appreciate it, and then I'll show you how you can replace the atlas with any arbitrary atlas that you might prefer. So the DKT atlas has a bunch of regions, and one of the first things that the code does is derive the mean value from your contrast map within each parcel. And next, it compares this regional activation profile, or face selectivity, with the expression profile of each gene using the partial least squares regression. You can also tell the toolbox to use a simple correlation if you'd prefer. Now, if you don't want to use the DKT atlas, you can get your own parcellation in MNI space, and you can use the AuBGen toolbox, which I'm showing you the name there. You can Google it, but it's another command which is relatively easy to execute, but it will take any atlas you give it that's an MNI space and output this gene expression by region matrix for you. And so you can replace the matrix in the code with your new one. You can point at the new atlas that you fed to produce that matrix, and the code will continue on its merry way. And the last part of the command tells the toolbox that you'd like to run the partial least squares regression, which is the PLS argument there. And you must specify how many components you'd like the analysis to include as it tries to maximize the amount of variance explained in the gene expression or the face selectivity variance. For this example, I simply chose 5. As you'll see in the output folder, this analysis very nicely produces these plots saved into the output path, telling you how much total variance has been explained. You could see that red dot and those dotted lines that are 5 components is explaining over 90% of the explainable variance, which is pretty good. And on the left here, yeah, the output folder will also show you how much each component explained in terms of variance. So once you actually execute this code, assuming that everything's correct, after a few hours, it will spit out in the output directory a large number of files, some of which the most important are these TSV files, which tell you which genes contributed to each component in the regression. So here I'm just showing you the first few genes from the first and fourth component. And you can see in the leftmost column of each matrix there, you have the name of all the genes that contributed to that component. And you can also see there's the raw p-value and the corrected p-value for that gene's association with your contrast map of interest. Now, there are 2 ways forward from here. One is that you can do an enrichment analysis to get a summary of what type of genes are involved in each component. You could use TopFun, which is a website. You could just Google it, where you can simply paste in the list of genes, and it'll tell you if there are any clusters of genes in there that belong to any sort of cellular function or structure significantly above chance. The second way forward is to rely on the gene enrichment analysis that exists inherently within this imaging transcriptomics toolbox, which I can show you how that one works here. So this toolbox will perform what is called a gene set enrichment analysis using a set of genes described in this paper by Lake and colleagues from 2018 shown at the top. But in that paper, they used a single cell sequencing technique to identify which genes are enriched in different cell types across the nervous system. So in this plot, each dot is a cellular nucleus with about 36,000 nuclei sampled from across the brain. And the enrichment analysis will assess if any of the genes associated with your contrast map overlap significantly with above chance with the genes associated with different cellular groups from the nervous system, such as excitatory neurons shown there in orange in the lower right, or oligodendrocyte genes shown up in the purple cluster in the upper left. But in the output folder from the command we just executed, you'll find many figures saved with one of these cell type clusters in its name, denoting if any of the genes in that group were enriched significantly in your analysis. For example, I'm showing you here the output for the enrichment analysis, asking if oligodendrocytes are present significantly above chance within any given component within the squares regression. So the top port of that, the top portion of that plot shows the running enrichment score in green. So for the gene set, as it walks down the ranked list of genes, and the score at the peak of the plot, the score furthest from 0 is the enrichment score for that gene set. And the middle portion of the plot, those blue vertical lines, shows where the members of the oligodendrocyte gene set appear in the ranked list of, of genes. And these genes are ranked in terms of how positively correlated they are with phase selectivity in the very bright red on the left, and how negatively correlated on the far right. And you can see the corrected p-value there would suggest that cortex that shows a preference for faces seems to be associated with a higher density of genes related to oligodendrocytes. So this is interesting and very cool. So this is a structure-function relationship that we discovered together just now. And one thing that's interesting is that we know face recognition develops with age, improving across childhood. And so does this mean that myelination in a face-selective region might develop more dramatically compared to a neighboring visual region that's not face-selective? So how could we test such a thing in the living brain? So this brings us to the next part of our cooking show, and how to use new MRI techniques to measure tissue environment across the brain in a living participant. So how this works is that in the MRI machine, which I've cartoonized here with 2 magnets, after a proton has been excited, which I'm showing you there, it's processing in a high energy state, but after some amount of time, it will relax back to field alignment. And we can measure that time precisely with actual units. So in order to measure this proton behavior precisely, you can collect a couple of different types of images from your scanner which we call quantitative MRI. So this particular protocol was developed by Aviv Metzer. Shown, his name is in the upper right, and a link to the GitHub repository is also there as well. But the first type of images that we'll collect in this protocol are EPI inversion recovery scans. These are relatively quick scans, less than 2 minutes each, and you'll want at least 4 of them spaced around these inversion times that I'm showing you here. There exist also rapid T1 EPI mapping sequences that give you more than 4 within the same amount of time as 1. I think MGH and John Polemeni has a nice rapid EPI sequence for that as well. So if you're interested in that, you can email him. But next, to precisely measure the relaxation of the proton, you'll need to collect spoiled gradient recall to echo scan of different flip angles. Generally, we use these flip angles of 4, 10, 20, and 30, depending on the resolution you want. These take about 5 minutes each for a whole brain image. So the SPGR scans let us estimate the relaxation behavior of the proton, and the EPI images let us estimate signal biases across the brain and remove those biases so we can precisely measure the relaxation time of the proton. And to do this, to analyze these 8 scans, you can download the Mr. Q toolbox or the MRQ toolbox shown in the upper right there. And you'll just need a simple MATLAB wrapper function to execute the Mr. Q run command at the very bottom there. Basically, this wrapper function just sets up 2 data structures, one telling the code parameters for the EPI sequence, like the TE, the TR, and the magnetic field strength and the flip angle for the SPGR scans. And then the Mr. Q run command, you just feed it the directory where the data lives, the data being these nifty scans, the output directory where you'd like everything to be saved, and then the two data structures detailing the EPI and SPGR parameters. And then once you execute it, assuming it runs and you have all the necessary toolboxes, it will, I think, complete in a few hours. And then in the output directory, you'll have lots of things, but presumably we're most interested in the brain maps folder produced in the output directory, which includes these whole brain maps of different tissue parameters. We'll focus just on two of them today. One of them is the T1 relaxation time. So this shows you at every voxel the precise amount of time it took the average proton within that voxel to relax back to field alignment, called T1 relaxation, or relaxivity R1, which is just 1 over T1. And you can see relaxation is very sensitive to myelin content. You can see its relaxation is very quick or low, where myelin is high. So it's very dark in myelin in those white matter voxels. And the great thing about qMRI is that it also lets us measure or estimate in every voxel the exact amount of the voxel that belongs to tissue and not water. So this is called macromolecular tissue volume, which is the TV map file shown there. And this is the precise fraction of the voxel that belongs to tissue. And importantly, these MRI scans give you brain maps, as I'm showing you here, with actual units. So they're not relative. These are numbers that you can meaningfully compare across people or across brain regions within an individual. And so remember that question we asked a few slides ago, if face-selective regions might be myelinating more dramatically than a neighboring face region than a neighboring visual region across development. So we can use qMRI to see if that might be true. So here, to cartoonize some predictions, I've just drawn two interconnected neurons, and so we can see what happens to proton behavior if tissue grows or is pruned with development. So if tissue were to grow in this region, then a higher energy proton, like a spinning top, would have more stuff to bump into and exchange energy with, and it would relax back very quickly. However, if tissue was getting pruned or lost with development, then the proton would be free to process longer for an uninterrupted for a longer period of time, and it would relax back more slowly. So in this case, pruning would predict longer T1, while growth would predict shorter T1 with development. So from some experiments I ran back at Stanford, we performed fMRI on children and adults to localize face and place selective cortex, which I'm showing you in red and yellow there. And then in the same individuals, we scanned them with qMRI to measure T1 relaxation time in the same voxels in the same participants. So I can produce a distribution of T1 relaxation times from the voxels of a subject's face selective region and average that distribution across participants. So here, kids are in light red, adults are in dark red, and what you can see is that there's a very appreciable decrease in T1 relaxation time across all voxels from childhood to adulthood, suggesting that there's widespread growth of tissue. And while tissue seems to be growing in the face selective region, as we hypothesized, we actually don't see any growth in the place selective region. So this seems to align very nicely from the transcriptome analysis that we just performed, suggesting that face selective regions have a richer density of oligodendrocytes and therefore might show more dramatic myelination across development. So here we've linked differences in function, face and place processing, to differences in structure in the living brain. So lastly, in this really brief last section, I want to go over how you can directly assess proteins themselves in brain tissue. So we assumed in the first section of our cooking show that RNA translates into proteins of interest. And with qMRI, we assume that it's sensitive to some of these macromolecules and proteins, but neither directly measure the protein content itself. And proteomics can do just that. And we recently applied it in a study of the cerebellum. So the cerebellum is separated anatomically into lobules, and it is posited that as you go from the brainstem towards the posterior cerebellum, that there is a sensory association gradient, with posterior lobules supporting more cognitive computations and anterior lobules in the brainstem supporting more sensory representations. And so I'm cartooning that here. Posterior lobules like CRUS1 and CRUS2 are shown in dark gray. They're suggesting they might have a more cognitive function. In a recent paper that should be published in a few weeks, we use the T1-T2 ratio from the HCP dataset, which is thought to be sensitive to myelin content. And we show that this sensory association gradient seems to be associated with a myelin contrast gradient as well. with anterior sensory lobules showing higher T1, T2 ratios, suggesting more myelin compared to posterior cognitive lobules. And you can appreciate that visually here in a 3D rendition of the cerebellum. So to see what proteins were potentially contributing to this MRI-based metric, we performed proteomics. So collaborating with Dr. Vayeni from the Children's Hospital of Philadelphia, we obtained a formalin-fixed cerebellum sample and extracted tissue samples from anterior sensory-based lobules 1 and 4, and samples more posterior in lobule crus 2. And these tissue samples were then prepped for mass spectroscopy to analyze the protein environment and assess how proteins may differ between these lobules. Most of this prep work can actually be done by the proteomics core at your university. There are also third-party vendors that you can just send formalin-fixed tissue to, and they will do this spectroscopy analysis for you as well. And it's relatively cheap. I think a given tissue sample costs less than it does to scan somebody in the MRI machine for an hour. So it's pretty, it's worth looking into. But the first step that the proteomics core or vendor might do for you is they homogenize the tissue sample, turning it into a cellular soup, and then they use trypsin to digest that sample and break it into smaller peptides. And once this has been done, these tissue samples are fed to a mass spectrometer to perform mass spectroscopy in an approach we call shotgun proteomics, because we're basically shotgunning all the proteins that are present in the tissue sample to see what it's made of. And equal volumes from different tissue samples go into the spectrometer. They become ionized and they're accelerated. And then based on their peptide mass and charge, they will land on different locations in a detector at the end of the spectrometer. And we can then identify which proteins are present within the sample and derive their relative abundance based on how many proteins were observed overall in that tissue sample. So, for example, like the protein, myelin basic protein, that we can determine what percentage of the tissue sample was myelin basic protein. And because it's a percentage, it's a normalized metric, so we can combine tissue samples across different lobules to compare them. So basically, once you get the data from your proteomics core, you can load it into a program like Scaffold 5 to see which proteins are present and to how much and to what extent. And a program like Scaffold can export all of your proteins and then how much was identified in each tissue sample into a CSV file, which then you can then analyze with MATLAB or Python to produce something like this. So once you have your CSV file, you can calculate, for example, the difference between the expression of each protein between your two regions of interest to produce what is called here a volcano plot. So this is a pretty standard mass spec visualization. And in this, each dot is a protein, and its location is determined by the log2 of its expression difference between your two regions of interest. And then on the y-axis, its location is determined by the significance of that difference, or the log10 of the p-value. And at a high-level glance, you can see that there's two spouts, which is good, suggesting that there are some proteins more highly expressed in lobule 1 and some proteins more highly expressed in crus 2. But we can threshold our data, so in darker green are those proteins that are significantly different between the two lobules. And some of the proteins I've highlighted there, but you can see, for example, protein like myelin basic protein, or MBP, is more significantly expressed in lobule 1 compared to crus 2. And you can do an enrichment analysis using TopFun, like I showed you before, feeding these protein lists. And what we find is that anterior lobule 1-4 has an enrichment of proteins involved in synapses, neurite growth, but importantly, myelin, verifying or suggesting that the MRI-based metric that we showed with T1, T2 ratio, suggesting that anterior lobules might have more myelin, seems to mirror this proteomic analysis, while posterior lobules seem to have genes involved in other things like DNA regulation and astrocytic development. So these findings offer more evidence at the level of individual proteins that the tissue environment in functionally distinct regions of the cerebellum are structurally distinct. So to summarize, I introduced some software packages and pipeline examples to analyze transcriptomics, quantitative MRI, or even proteomics to relate structural differences between brain regions to differences in their function in either the living or postmortem brain. I hope you enjoy HBM and Glasgow, and have a pint for me. Thanks.